The cell cycle has two kind of major phases to it. The first phase is interphase, and as you can see on this diagram here, interphase really makes up the majority of the cell cycle. So this whole area here is our interphase. And during interphase, a cell is basically just being a cell. It's doing what a cell is supposed to do. It's gathering nutrients, it's making energy, it's performing a function for the body. Maybe it is an immune cell and it's killing off pathogens, or maybe it's a muscle cell and it's contracting in order to allow something to move. During interphase, DNA is also replicated when it needs to be. And this is important because in order for a cell to divide, it first has to make a copy of its DNA, because you have a complete set of DNA in every single cell in your body. So in order to take one cell and split it into two cells, you have to take one set of DNA, make a copy, so that then each new cell gets a complete copy of that DNA. So you can see that DNA replication happens during this DNA synthesis phase here. The second part of the cell cycle is this mitotic phase. And the mitotic phase actually makes up a relatively small period of time in the cell cycle. So during mitosis, what happens is that the DNA that was replicated during interphase gets separated and divided into those two new cells that are being made. In addition to the DNA getting separated, all the stuff in the cytoplasm also gets separated. So that means that these two new cells that are being made, so basically we've got one cell getting divided into two cells, and in those two new cells, whatever DNA is in one cell, each new cell has to get a set of DNA. They also have to get things like mitochondria and ribosomes and the endoplasmic reticulum and cytoplasm. And so all of that stuff has to be copied and then divided into the two new cells. Now, those two major phases that we talked about are further broken down into kind of smaller phases. So remember that interphase is basically this whole area of the cell cycle here. But we can see that there are actually three kind of subphases that go along with interphase. So the first one is actually the G1 phase, followed by the S phase, and followed by the G2 phase. So you notice that I skipped over this G0 phase here. And I did that on purpose because you'll notice that in the diagram here, the G0 phase is not on there. And so the G0 phase is actually kind of like an offshoot of the cell cycle. And we'll talk about what that is and why it matters on a future slide. Now, the mitotic phase, which includes mitosis and cytokinesis, also includes the subphases of mitosis. So mitosis we can kind of abbreviate this as PPMAT, and that stands, or that this mnemonic helps you remember the order of the events that happen during mitosis. And so the first part of mitosis is prophase. The second part is prometaphase. The third part is metaphase. The fourth part is anaphase. And the fifth part is telophase. And then the second main part of the mitotic phase is known as cytokinesis. Now, the key difference between mitosis and cytokinesis is that during mitosis, you have the nucleus dividing, and you have the chromosomes, which, remember, the chromosomes are housing the DNA. So the nucleus and the chromosomes divide. And then during cytokinesis, everything else divides. So during cytokinesis, the cell membrane pinches in, to divide the cell into two smaller cells. And each of the new cells gets one of the new nuclei that was created during mitosis It also separates things like the cell membrane. The cell membrane gets kind of chopped in half, and the cytoplasm and the organelles get divided in half so that each new cell being produced, which is called a daughter cell, the new daughter cells are getting a new nucleus full of a complete set of chromosomes, plus they're getting cytoplasm with organelles like mitochondria, endoplasmic reticulum, Golgi apparatus, ribosomes, etc., etc. So now let's talk about that G0 phase that I kind of skipped over on the previous slide. So the G0 phase is kind of an offshoot of the cell cycle, but we still talk about it with the cell cycle because it is an important part of what a cell goes through during its life cycle. Now the G0 phase is also called the quiescent or inactive phase. But I don't really like these terms because it makes it sound like the cell isn't doing anything, and that's not the case. It means that the cell is not dividing and it's not preparing to divide, but it's still doing what the cell needs to be doing. So the cell is still active with regards to it's making ATP. It's performing functions for the body. So, if it's a muscle cell, it's still contracting to move the limb or the body around. If it's an immune cell, it's still attacking pathogens. So it's not inactive. It's not just sitting there doing nothing. It's still being a cell and it's fulfilling its role as a cell. The only thing that's inactive about it is that it's not preparing to divide. So it's not preparing for mitosis. So mitosis is not in the near future. Now this G0 phase does not occur in all cells. So some cells never go into the G0 phase, and so they're constantly replicating. Some cells go into the G0 phase temporarily, meaning that they might stop preparing to divide for a little while until they get the signal to start dividing again. And then some cells go into the G0 phase permanently, meaning that once they are produced, they will never divide again. And that's true of things like nerve cells and mature cardiac muscle cells. And this is one reason why things like nerve damage and damage to the heart muscle can be so serious, because once these cells mature, once they're produced and they mature, they don't go through mitosis again, so they can't heal and regenerate. So something like a heart attack will have long-lasting effects because some of the heart function will be lost due to those cells dying. So now let's take a look at the subphases of interphase during which a cell is preparing to go through mitosis. So for cells that are preparing to divide again in order to make new cells. So the first stage that we're going to talk about is the G1 phase. And you can see that here in interphase. All right, so this is the first phase. And the G for the G1 phase technically stands for gap, but I like to think of it as standing for growth because this is a time of growth for the cell. So the next phase that's going to be happening is the S phase, which is DNA synthesis. So think about the cell is growing and preparing to make DNA. So you can imagine what it needs to do in order to prepare to replicate DNA. So during this first gap phase, the cell has to get nucleotides because in order to make DNA, you have to, remember, you take the original double strand of DNA, you pull it apart, and then you add new nucleotides to the existing strands in order to make the new strands of DNA. So you have to have new nucleotides in order to make new DNA. It's accumulating and making proteins, for example, enzymes. Remember that there are a lot of enzymes involved in DNA replication, and there are proteins involved in organizing the DNA into chromatin. It also accumulates energy reserves, so it might accumulate different nutrients so that it can then make ATP, because DNA replication requires a lot of energy to occur. So it's going to gather things like nutrients and oxygen to make ATP so that the DNA can be replicated in the next phase. The second phase of interphase is the S phase, which stands for DNA synthesis. So remember, the first phase was that G1 phase. Second phase is the S phase, and that S stands for synthesis. Remember that synthesis means to make something. And so this is also the same thing as saying DNA replication. So this is just the event in which you had a double strand of DNA that divides, and then each original strand gets a new strand of DNA added to it. So we learned about DNA replication in a previous lesson. So if you don't remember what happens during that, you should probably go back and review what happens during DNA replication. So as I mentioned on the previous slide, the S phase is where DNA gets replicated. And remember that in eukaryotes, and eukaryotes are cells that have a nucleus, which includes humans. And so in eukaryotes, the DNA is packaged in these structures called chromosomes. And more specifically, these chromosomes are linear, which means that they look like a bar or just a line. And you can actually see that on this picture down here. This is called a karyotype, which is just a picture of chromosomes that have been removed from a cell so that we can study them and look at them. And so you can see that these chromosomes are these bar-shaped chromosomes here. So recall that these chromosomes are just made up of this kind of double helix of DNA. And remember that in humans, we have about 3 billion base pairs in our genome. So we have a lot of information in our DNA, and this is a very, very long molecule. So it needs to be organized to keep it from getting tangled so that the cell can actually access the genetic information that it needs when it needs it. So this DNA gets wrapped around these proteins called histones. And so this protein here would be a histone with that double helix of DNA kind of wrapped around it. Now, each histone with some DNA wrapped around it is called a nucleosome. And these nucleosomes get stuck together in this chain, forming this substance called chromatin. Now, chromatin and chromosomes are essentially the same thing. The only real difference is how tightly coiled they are. So chromatin is kind of like a loose string of these nucleosomes stuck together, whereas a chromosome is further condensed, meaning that the chromatin is kind of coiled or wrapped around itself to make it a even more organized structure. So what we're seeing on this karyotype over here, these are chromosomes. And prior to being chromosomes, they were just chromatin, which is just a looser molecule where it would just be kind of more like stretched out and stringy. Now, humans have 46 total chromosomes. So we have 46 of these bar-shaped chromosomes, but it's better to think of them as being in the form of 23 pairs of chromosomes. And you can see on the karyotype down here that we have these pairs of chromosomes. And so rather than saying we have 46 chromosomes, we say that we have 23 pairs. And the reason why that's important is because we essentially get 2 copies of genetic information. We get 1 copy of genetic information from our mom and 1 copy of genetic information from our dad. And so your genetics are really determined by the combination of your mom's chromosomes interacting with your dad's chromosomes. And so basically, if we answer this question, where did these pairs come from? They really come from sexual reproduction. So approximately 9 months before you were born, your parents had sex, and that's how they made you. And so what happened is that your mom had an egg, and that egg had 23 of these bar-shaped chromosomes in them. Your dad had a sperm that also had 23 of these chromosomes. Well, the sperm fertilizes the egg, which ultimately forms something called a zygote. And the zygote has these 23 pairs of chromosomes in them, or that's where we get the 46, because 23 times 2 is 46. So if we come back to our karyotype picture here, one chromosome in each pair came from mom, and one chromosome in each pair came from dad. Now it's important to note that these paired chromosomes are not identical to each other. They're similar in the type of information that they carry, but they are not identical. And we'll talk about that more when we get to mitosis and meiosis. So on the previous slide, I mentioned that in eukaryotes, the chromosomes are these bar-shaped chromosomes. And that is true, but sometimes the chromosomes actually take this kind of X shape to them down here. So what's the difference between the bar-shaped chromosome and the X-shaped chromosome? And the key difference there is what happens during the S phase of interphase of the cell cycle. So the bar-shaped chromosome, you can think of this as like a package of information. And when the DNA gets copied, when it gets replicated, what happens is that the two copies get kind of like stapled together or stuck together. And so when we end up with this X shape, what we end up with are two copies of the same information that are just stuck together. And this is going to be temporary because eventually these two copies are going to be separated, and each new cell that's formed during cell division is going to get a copy of the original information. Now, once these chromosomes are X-shaped, each one of the arms is called a sister chromatid. So in this picture here, we've got a sister chromatid on the right and a sister chromatid on the left. And these two copies are identical. So these sister chromatids are identical to each other. And this is important because each new cell that's going to be created during mitosis is going to get an identical copy of the original information that was in the original cell that decided to divide. And after these sister chromatids are produced, they're actually connected to each other. So I mentioned that it's like making another copy of the information and then stapling it to the original copy. So they're stuck together for a while until they're no longer stuck together, which we'll talk about when they get torn apart a little bit later on. And this region where they are connected is called the centromere. And there are actually some kind of special binding regions called the kinetochores. We'll talk about that more when we get to mitosis. Now, if we look at this picture over here, so this is a karyotype, which is just a picture of the chromosomes that have been taken out of a cell of an organism that we're studying. And you can see that these two karyotypes are similar but different. And the main difference that you should be seeing is that we still have those paired chromosomes. And remember, we get those paired chromosomes because of sex, right? So paired chromosomes happens because your parents have sex and you have a sperm that fertilizes an egg, and you end up with two copies of every single chromosome in your body. Now, the main difference actually has to do with whether these chromosomes are linear or X-shaped. And so you can see that on the left-hand side here, we're looking at before the S phase. So this is before the DNA has been replicated. And so each one of these chromosomes is just made up of a single bar shape to it. All right, and they're not attached. They are paired up, but they're not attached to each other by that centromere region. Now, after the S phase, so remember that during the S phase, you've got your DNA replication occurring. And so the bar-shaped chromosome, a copy has been made and it's been connected to it. So these chromosomes have more of an X-like shape to them. And you can see that really well down here if you look at chromosome number 20. You can see that really characteristic X shape versus number 20 over here, which is just showing you two lines. All right, so this can get confusing, so let's do a quick recap. So remember that before DNA replication, so before the S phase, each chromosome is only made up of one chromatid. So we've got, you get one chromosome from your dad and one from your mom. And so in a human cell, you would see 46 chromosomes, and each chromosome would just be called a single chromatid. After DNA replication, so after the S phase, these linear chromosomes start to look like an X. And that's because each chromosome is now made up of two sister chromatids. So this is still considered one chromosome, but it's made up of two sister chromatids. Now, these sister chromatids are identical, and that's because one arm is literally an exact copy of the other arm. So each one of these chromatids are identical. Now, we still have our paired chromosomes. So here we're just looking at one pair of chromosomes. Whereas remember that in humans, you would actually have 23 pairs for a total of 46 chromosomes. On the right-hand side, we still have those paired chromosomes. We just now have two copies of each of those chromosomes. So we're still talking about interphase, which remember interphase makes up this entire part of the cell cycle. We already talked about the G1 phase, which is our cell growth or our first gap or growth phase. We just got done talking about the S phase, which is our DNA synthesis or DNA replication phase. And then the third phase is another growth phase. So we call this the G2 phase. And it's actually the third phase of interphase. And during the G2 phase, the cell is just preparing for mitosis, all right? So it's replenishing energies, all right? So it's going to gather up things like nutrients and oxygen so that it can make ATP. And it needs ATP because mitosis is an active process. It takes a lot of energy in order for it to occur. We also need things like proteins in order for mitosis to occur, and we'll talk about what those proteins are. So you can just think of it as another kind of growth and preparation phase to get the cell ready for the mitotic phase.